Hello friends, I welcome you to Mannan's e-learning program. This lecture is in continuation of my previous lecture, how to calculate the recombination frequency, and I also explain in that lecture about independent assortment dihybrid cross ratio. I introduced the concept of genetic map over there, so you can visit the link and you can get benefited from there. So I will be discussing in this session about the genetic and physical map. Now if you see in terms of the words which is used here, one is genetic and the second one is the physical, and the third word where which is used as a map. So if you go with a map definition, like now you know that there is a Google map which is present, which can give you direction. Google Earth is there where you can see the exact location and a picture. So on the same way, when we are interested in knowing the order of a gene, or we are interested in knowing the exact base pair difference between a gene, difference, or I would say the distance between a gene, then we use a genetic and physical map. Both of them has their own advantages, and it is used, you can say, for sequencing experiment. Okay, sequencing experiments, when I say this is not the exact sequencing I am referring, I am actually referring to the genome annotation. So what you can expect in today's session, that what is the difference between a genetic map and what is a physical map. The genetic map is usually used based on the recombination frequency. I will briefly talk about that how it can be calculated, but for more details I will suggest you to visit another lecture which I will share the link. It is a 45 minutes lecture, but you can get benefited and clear your concept from there. Now let me show you a picture. In this picture, if you see, there is written a genetic map, a cytogenetic map, a physical map, and a DNA sequence which has been shown. Now genetic map, there are certain numbers which has been written, and it is shown also as a centimorgan. The centimorgan is based on the genetic map is one of his student, Alfred Sturtevant, who is the first person to create a genetic map. He was in his 20s when he basically drew that map, and because of his work in the Morgan's laboratory and the guidance which he received from the Morgan, that's why we call them as a centimorgan. So 1 centimorgan is equivalent to 1 map unit, or in short it is also called as map units, and it is also equivalent to, you can say, 1% recombination frequency. So that's how you, if you know or you can able to calculate the recombination frequency, then you can able to draw a genetic map. Usually in genetic classes, the faculty members or the teachers, they explained about 2-point test cross or 3-point test cross, like this type of terminology you come across. So when they referring to a test cross, so this definition I also explained in one of the very early lectures in genetics, that what is the difference between back cross, what is test cross, what is reciprocal cross, and others. Just to refresh the memory here, when we mate two plants, okay, like P1 plant versus a P2 plant, we basically get an F1 progeny, or the first filial generation progeny. Usually for Mendelian experiments, you use a dominant phenotype of a plant, and one is the recessive one. So let's say take an example here, say purple flower color plant is crossed with a white flower color plant. So in F1 generation you get a heterozygous population, all will be purple. This F1 or the first filial generation plant is crossed with the recessive parent, that is called as a test cross. So this is for one gene. When you add two genes, let's say seed shape as well as you can say the flower color, then that becomes a 2-point test cross. Means you are whatever the F1 progeny which you are getting from this particular cross, and most likely which I am writing here also for simplicity's sake, that it will be heterozygous plant, and when it is crossed with a recessive plant, then that becomes 2-point test cross. And similarly for a 3-point test cross, when you add up another character or another gene, say a round seed shape, the purple flower color, and say a height of a plant, then that will becomes a 3-point test cross. And then for heterozygous, then you can also refer to here. If you are not clear with this concept, then I will suggest that you visit my lecture in which I have explained about independent segregation, the Mendel's first principle, and I also explain about Mendel's principle of independent assortment. What is monohybrid cross, what is a dihybrid cross, how you calculate this, that you can get clarified. So I will skip this part here, and so let me rub this, and then I shall focus here about the genetic map and a physical map. What is its difference? So I just said that it was developed by the Alford, a Morgan student, and this is Centimorgan. What is his definition? These numbers which you are seeing, 20, 30 or 30 something like this, these numbers basically denotes the distance between the two genes in terms of centimorgans, which are calculated based on the test cross. In case of a cytogenetic map, because this picture is also talked about, so you see here there are certain dark color bands and there are certain light colored bands. Usually for a chromosomal staining, we use certain strain. One dye or a stain which has been used is called as Giemsa strain. The cytogenetics has its own importance. It can tell you about the heterochromatin and the euchromatin regions. The heterochromatins are those regions which are densely packed, transcriptionally inactive region, and similarly a euchromatin which is transcriptionally active region. It's mostly taught in case of the discoveries of transposons and cytogenetics. So maybe in future I will discuss about this. Let's focus here only in terms of genetic and physical map. So the physical map, as you can see here, there are again certain numbers which is been shown, and also the units which is been shown here is megabases. Okay, now if you take about, say for example, human genome, the human genome size is roughly 3 billion bases, okay, or 3000 million bases. That is a human genome size. Similarly, if you take another plant like Arabidopsis thaliana, its genome size is roughly 121 megabase pair. If you take a E. coli, its genome size is roughly around 4.3 megabase. So there is several genome sequence projects are there, like rice genome is already been known, wheat genomes is almost complete, Similarly, zebrafish genome is known, Drosophila genome is known, several of other animals or close relatives of humans like chimpanzees genome sequence already done. So like that. Now all of this, where we are interested on knowing the exact distances between the genes, then we use a physical map into consideration, and the technique which is used is the sequencing technique. Okay, the sequencing, the direct sequencing, or you can say the Sanger sequencing, which was initially used for sequencing human genome. Nowadays you will find that people now use a next generation sequencing, like for example Illumina platform, or in some places they use still the pyrosequencing over there. That requires a different discussion. Maybe in a molecular biology lecture I will describe about that as well. So let's take another look about a genetic and physical map to make you give a more clarity here. So if you see here in this particular picture, let me change my color code. This is the orders which has been shown on a particular chromosome. This is called as genetic map, and we are knowing when the exact sequences, and this sequencing which you do, as I said, by Sanger sequencing initially, which is one done, you called as a physical map over there. The techniques which is been used is beyond today's class discussion, but essentially, as I'm saying, it's involved the sequencing, and for genetic map you basically use the test cross and you find out the recombination frequency, and based on recombination frequency one can say about what is the genetic map. I can, to make you more understandable, I can give you an analogy that in case of genetic map is like a paper diagram, okay, or a paper map where people used to use when there is no GPS or no Google Maps is available. Physical map is more like a Google map or any GPS device where you just put out the coordinates and then you reach to the exact location. With a paper map you still also reach to the location, but to close to that location. So both of these maps give you an information about the genes which are located on the chromosome, their coordinates in the chromosomes, their order in their chromosome. And why that is important? Because if you want to study a particular gene, you should know its sequence, and you should also know the function of the gene. Now so like for example in physical map, so physical map give us the exact location, okay, so that is its advantage, but it cannot give you an information about the function unless a experiment has been done on that. However, in genetic map, where you cannot find out the exact sequence of the DNA, but you can all find out the order of the genes and a relative distance between the gene, and you can also get a more information in terms of a functional aspects, if certain mutants which is been used or certain other techniques like RAPD, RFLP has which is used in case of genetic mapping. So both are important, okay, and both are used in genome annotation. Now if you see this picture, which is a comparatively a very complex picture, but that's how it looks like. And to give another same analogy which I am using here about a Google map, so you can see this is like a satellite image of a particular town. We do not know, maybe in that particular town we are interested in knowing intricate details, then we have to see a particular distance between a gene. We may be interested in knowing the distance between a particular gene, but if we can get a bird's eye view that what is been present, okay, so that's how this looks like. This is about a chromosome 1 which is 88.7 MB which has been shown. This is all are the genes and its coordinates, as you can see over here. So that's how it's looks like. Now one of the important way to find out, like because we will focus our discussion for only genetic map here. This genetic map, if you get some idea, you can also know that what is their distance. Like for example, here it has been shown 3 genes, and by the way these genes are referring to the Drosophila. So this is, and it was taken from the Alford's work, which is having a more detailed genetic map. But if we know a distance between 2 genes, like say for example 3 genes, that is Y, W and M. If I know the distance between Y and M, which has been shown also here, 35.4 centimorgan, okay, and the distance between these two, that is Y and W, is 0.5, then I can easily calculate what could be the distance between W and M. The simple way to do is you can take 35.4 minus it with 0.5, that's give you a value of 34.5, as you can see here. So a estimate can be made based on the recombination frequency. It also tells us that these 2 genes, that is Y and W, is very close apart, means they are very close. So there is a chances that these genes could be linked genes. However, these 2 genes Y and M, or W and M, they are far apart, so the crossing over event can take place. In fact, this is this experiment leads to Morgan his classical experiment of sex-linked inheritance in Drosophila eye color from this observation, and he basically gave a whole detailed explanation based on this. And his experiments, along with the other experiments which he did, he was awarded a Nobel Prize. So that's kind of information you can draw based on the genetic map. How to calculate the genetic map? As I said, it can be calculated based on the recombination frequencies. How you calculate the recombination frequency? For detail, you can visit to this link, and then you can get benefited from this. But essentially, one has to calculate the number of the recombinants by the total number of the offsprings multiplied by 100. So in any particular cross, like say this is a red flower, a round seed shape, and a yellow cotyledon color cross with a wrinkled seed shape, a green cotyledon color in F1, you get a heterozygous population. And when you cross this with this parental 2, let's say, and this is parent 1, and you calculate the total number of parental population, okay, you basically get one parental population, that is P1, then you basically get P2, you get R1 and R2. This population you can calculate that how many recombinants which you are getting by the total number. You can say what is a distance between R and Y genes, and you can exactly apply this to a 3-point test cross, means 3 genes into consideration as well. So that is how you calculate a genetic map and a physical map. There is one more thing, and then I will basically stop here, that if you see in this particular examples, the letters which denotes, let's say, these genes B, A, D and C, if you know the distance between B and D, as you can see here also shown, is 40%. So this 40% is basically referring to the 40% recombination frequency. So it will be exactly 40 map units, or you can say 40 centimorgans. That's how you define them. Now when I know the distance between B and D, okay, and somehow if I know the distance between A and D based on the 3-point test cross, which is, let's say, 25%, or we can say it's 25 centimorgan, we can calculate what is the distance between B and A. It is simply addition subtractions which we use. So B and D we know it is 40, and A and D we know it is 25. So distance between B and A, if I have to calculate, I will simply do a math. 40 minus by 25, so it comes around 15. So I will write down the distance between them as 15 centimorgan, 15 centimorgan. And if you do not know, let's say, this information, somehow you know information what is distance between B and A, you know information what is distance between A and D, based on the recombination frequency, you can always say what is the distance between B and D. It will be simply 25 plus 15, and that makes it 40. That's how you can, that is the beauty of, you can say, the recombination frequency, that you can find out the gene order. You can also find out the distances between the genes. So let me summarize in today's lecture or today's session what I mentioned. I mentioned about that what is the definition of genetic map, what is the definition of physical map, what is the implication of physical map and genetic map, because both are required for genome annotation. You cannot predict the function based on physical map, you still require a genetic map to know the function. Both are used in combination to find out the exact picture of the gene function and its order. Why we are interested? Because we are interested in knowing the gene, so that we can clone that gene, we can mutate that gene, or you can express in some other system and do whatever the information we want to do. I also explained to you that how it looks like in a bird's eye view. I also explained to you that how you can calculate, not in example terms, but at least a formula way. And I also explained to you that these are additive, that is, and you can predict the gene order. With this I will stop. If you like my video, I will request you to please subscribe to my channel. Thank you very much, and see you in another series of lecture. Thank you.